So again, welcome to the March 6th instance of the IMAG MSM Working Group meeting. Just have a few announcements here, and then I'll turn things over to Chase Cockrell from the University of Vermont, who will be talking about the wound environment, agent-based model, a digital twin for wound healing. Just as a reminder of the people, this group is run by Tom Helikar and James Glazier, with web administration by James Sluka and Lorenzo Vescini. If you have any questions or would like to submit any speaker ideas or anything like that, please feel free to contact any of them. We can try to get it on the schedule. Just a reminder of our social media links. We do have a group Slack channel, our Twitter/X profile, wiki page, LinkedIn, and YouTube. Please do feel free to participate in those, and if you have any announcements you'd like us to send out through our social media, we're happy to do that as well. Again, if you can also feel free to retweet our announcements on X, helps us get a little bit more participation here and there. And if you have any publications that you'd like to share, we can publish those actually to the IMAG MSM wiki. So please just send those to jimsluka@iu.edu, and we can get them hosted on our page, any recent publications that you or any of your students or colleagues may have put out that are relevant to the IMAG MSM topics. As a reminder, the GLIMPRINT website can now host job announcements. So if you have any new job announcements or announcements for workshops, meetings, publications, or funding opportunities, please feel free again to send those to Jim Sluka, and we can get those hosted through the GLIMPRINT site. A couple of workshop announcements. We have a new workshop this year to announce, the cytoskeleton modeling workshop. This is run by Julia Belmonte at NC State University. That'll be from May 12th to 16th. The registration deadline is March 30th. And I will be posting all these links in the chat once I finish rifling through the various announcements here. A couple of competition announcements. We've got our OpenVT 2025 reproducible agent-based model competition. The deadline for those submissions is March 31st. And we also encourage people to submit their models to the Biomodels Model of the Year competition as well. If you have any questions about the OpenVT reproducible modeling competition, please feel free to email openvtprize@gmail.com, or any of the coordinators here can point you in the right direction. We also have a multicellular modeling workshop at the beginning of the SMB meeting for 2025. This will be a one-day workshop in advance of the conference on July 13th. And if you'd like to sign up for that, we do have a Google form. Again, I'll be posting the link in the chat here in just a minute. One last announcement. We've got the 4th International Summer Institute on Network Physiology. Application deadline for that coming up March 15th, so pretty soon here. If you or any of your students would like to participate, we do encourage you to go ahead and get those applications in. One job posting that we have currently, the Moffitt Cancer Center is looking for a faculty member in mathematical oncology, and you can find more details here at the link, which again, I will post in the chat. Sorry, just a quick reminder of upcoming seminars. March 20th, we have Louis Gall from Birmingham University on mathematical modeling of drug-induced injury in the intestinal crypt. Then on April 3rd, we have Ramon Ortiz Catalan from the University of California, San Francisco. Again, if you would like to suggest speakers, please feel free to use our Google form or contact any of the coordinators here. Also, feel free to suggest yourself if you have something you'd like to present, even if you've presented before. We always like to hear from people, again, about updates on their work. Or if you have any students who are currently doing things that you think would be relevant to this group, feel free to suggest them as well. Any announcements? Quick announcements from anybody before we turn things over to Chase?
Oh, I'll just add for job opportunities, my group is looking for multi-scale modeler at the staff scientist level.
Okay.
Feel free to email me if you are interested or know of anybody who's looking for a job, please.
All right, so feel free to drop anything else in the chat as we go here, but I will go ahead and turn things over to Chase so you can get started on what I think will be a very interesting talk for today. So Chase, you should be able to take over the screen share now.
Well, thanks for inviting me to talk today. So I wanted to discuss our digital twin work in the context of volumetric muscle loss injuries. And today I kind of wanted to talk about everything we considered to build the digital twin, as well as, discuss and highlight the difficulties we had, both in terms of actually building the digital twin, but also in collecting the data and managing the expectations of our experimental collaborators. So the project that I want to describe, it's been my main focus for the last few years, and that's my participation in DARPA's Bioelectronics for Tissue Regeneration program. So the purpose of the program is to develop technologies that will return wounded soldiers to their full functional capacity after significant injuries. And the class of injury that our group specifically is looking at here is called a volumetric muscle loss injury, which, is just like it sounds. It's when the patient loses a significant volume of muscle. So it's an extremely debilitating injury, mainly due to the nature of the healing. So in a VML injury, VML for volumetric muscle loss, the biological imperative from years and years, millions of years of evolution, is to close the wound as quickly as possible to prevent contamination from the outside environment. And the solution that nature has arrived at, or that mammals have evolved to, is to rapidly fill the wound with collagen, which is then over time remodeled into fibrotic scar. And that's not functional tissue. And so in a lot of cases, it can even lead to amputation, as it did with the patient that you can see on the right of the screen there. And he was attended to by Steve Badalak, who's the PI of this DARPA project. And he's currently at the University of Pittsburgh. So the primary goals of the program are to alter the healing phenotype from the formation of fibrotic scar into the regeneration of healthy functional muscle, and also to increase the speed of wound closure by 50%. So those are the program goals put forth by DARPA. And a secondary consideration is to use the device in amputations to prevent wound failure. Sometimes after amputation, the wound just won't remain closed, and then the tissue loses its integrity and could even require a further amputation. So for example, if someone gets a below the knee amputation, that's something that a person can really learn to live with. But if the wound fails, a below the knee amputation can turn into an above the knee amputation, and that's much less functional. So on this slide, I briefly want to go over the process of muscle healing and what we want to do instead. So on the far left, you can see there's a bunch of muscle and it's been injured by some volumetric muscle removal. And so what the muscle really is a bunch of muscle fibers bound together or bundled together. So in this case, some of those fibers are still intact and some of them have a middle segment removed. And when the muscle is injured like this, chemical signals propagate around the area of the wound, and these signals activate what are called satellite cells, which are undifferentiated muscle stem cells. And these satellite cell reservoirs are located on each muscle fiber. So when the cells get activated, they migrate along the muscle fiber from which they originated. I'm on a little bit of shaky Wi-Fi channel, so I'm going to leave the video off for now. I'm on hotel Wi-Fi.
Okay, no biggie.
Sorry about that. So when they reach the damage, they fuse to the damaged muscle fiber, and ideally, then the muscle fiber is rebuilt up cell by cell, kind of in a linear progression from each end, and it meets in the middle. However, as I mentioned earlier, there's a competing process to muscle repair, which is the wound closure. So when this wound sends out signals that activate the muscle repair cells, it also sends out signals that activate a whole host of other cells, most notably fibroblasts. So in reality, the fibroblasts come in, they start secreting collagen, and then after the collagen has been secreted, it starts being remodeled into fibrotic scar. So one of the principal consequences of this introduction of collagen to the wound environment is that it sort of caps off the muscle fibers such that no binding sites are available, and thus the muscle can't repair. It can't build up cell by cell because that's blocked. So the void then fills up with collagen and scar instead of functional tissue. So as I've alluded to, it's a very large project, and I've got an overview of it on the screen here. So we start with a VML wound. A bed of sensors, which are developed at Carnegie Mellon and Northwestern, is then laid over the wound. And these sensors are supposed to measure specific molecular mediators in a spatially explicit fashion, and then pass that information onto a trained neural network, which is what we develop at UVM. And the neural net then controls an array of lasers which shine on genetically engineered cells, which are engineered also at Northwestern and Rice, to create a specific protein in response to certain wavelengths of light. And these proteins are then fed back into the wound at specific locations, informed by the neural network to control the healing. So my work here, and what Gary also worked on, is the development and training of the neural network controller. So in order to do that, the first thing we do is develop a mechanistic computational model of the wound environment. We then determine all the possible parameterizations, or a comprehensive number of parameterizations of the model that cannot be invalidated by experimental data. So it's not to say they're true or correct, it's just that with the data we have, they can't be invalidated. So then we use deep reinforcement learning across some of the more extreme boundaries of that parameter space to operate on the model and derive the optimal therapeutic policy for a heterogeneous population. So in order to get the data that informs the model, we're using a canine model of volumetric muscle loss and collecting a really wide range of data. So briefly, the wound is created in the animal, and then it's sampled at various time points throughout the healing process from day 0 to 42 days after the healing process. The sampling method used here is an excisional biopsy, so that does limit the resolution of the time series that can be obtained from any individual animal, because the biopsy procedure naturally disturbs the wound environment and the wound healing. So, as with almost all biomedical experimental platforms, the time series are constructed from a number of replicate animals that are sampled at different time points. So on the screen, you can kind of see examples of 2 wounds. On the left, you have one that's very early in the healing process, and you have a little schematic of where we would take the biopsies. And on the right, you can see a wound post-biopsy, and also see that wound is much further along in the healing process. So from those biopsies, we get gene expression and proteomics from Luminex, so we get protein concentrations. We also have some quantitative histology from biopsies that are adjacent to the biopsies that are measured for gene and protein expression. We also have some measures of innervation and electromagnetic stimulation, which measures any regeneration of functional muscle. And then to begin and to measure the healing process throughout, the animals are periodically imaged with a CT scan to give a full 3D view of the wound. So the WABM, or Wound Environment Agent-Based Model, represents individual cells as independent agents that act according to a predefined rule set. And there are a variety of cells represented in this model, including T cells, macrophages, neutrophils, fibroblasts and myofibroblasts, and muscle cells, all included at various stages of their development. The model right now is fully 3D and operates on a cubic mesh with voxels measuring about 100 microns on each side, which is the approximate diameter of a German Shepherd quadriceps muscle. And the model represents a wound that's approximately 4 centimeters by 3 centimeters by 2 centimeters, with several centimeters of healthy tissue outside of the immediate wound. So in this model, the main drivers of behavior are diffusible proteins. And when a cell encounters these proteins, it then makes new proteins in response and secretes those into the environment, or it performs some predefined function like move, chemotaxis, die through apoptosis or necroptosis, deposit collagen, fuse to a muscle fiber. I mean, anything biologically relevant to this process. So on the screen here, we've got a wireframe schematic of the interactions and relationships represented in the model. And each one of the relationships is a fact that has been taken from the accepted biomedical literature on muscle function and regeneration in mammals. So we basically read all the literature, or much of the literature, about volumetric muscle loss and muscle healing, and we make sure to include the pathways and the features that are common in all those papers. So what the community thinks is most important, that's what we model. And then we quantify those relationships with the machine learning techniques that I'll be discussing. So one thing that's vitally important to note in our philosophy, and should be apparent when you see the experimental data, is that there's no unique solution to this problem. So there's no one model parameterization that can encompass or explain all of the experimental data that we see. And so it's a bit of an alternate approach to traditional mathematical biology in that we're not trying to fit to the mean of some behavior to explain globally what's going on. Rather, we're trying to encompass the entire space of behaviors such that we can design a generalizable, or the most generalizable therapeutic policy for this specific injury. And it's not really surprising, I think, to do it that way because no two of us in this Zoom call are parameterized in the same way, referring to our genomes. And even if there were identical twins in this call, which I don't think there are, epigenetic effects could still affect and still generate different behaviors in their gene regulatory networks. So to instantiate the model in a realistic way, we start with the CT scan data taken immediately after the first surgery. So then we read that in and approximate everything below the surface to be healthy tissue, and we model the surface to be significantly inflammatory, which is a true statement. And we then evolve the model forward in time and generate realistic behaviors for what's represented. So I guess what I mean by that is overall tissue composition of the wound and spatially explicit protein concentration. So unfortunately, because we don't explicitly represent swelling and edema, the time points between the start and the end of the volumetric filling don't really match well on appearance alone with the animal model. So while we have good spatial distribution of the proteins and spatial distribution of the tissue compositions, in the intermediate stages, our generated wounds don't really look the same because we don't have any swelling. And I've got a few slides later that really illustrate that and some of the difficulties that it causes. So briefly, this is an extremely basic explanation of protein synthesis and sort of just how we represent it in the model. So I'm skipping over the details here, but very briefly, the gene, the segment of the DNA that's copied into mRNA is translated into protein, which can be secreted or internalized by the cell. So we take this basic machinery and put it in each cell of the model. So you have thousands, or actually you have millions of these sets of differential equations running in parallel that generate the gene expression profiles and protein concentrations seen in the experimental tissue. So for each cell, then, there's a set of coupled differential equations, and the differential equations we use are delay differential equations, meaning they operate on a time delay that link gene expression with protein production. And here you've got N being the number of genes, R being a vector of mRNA expression levels, P being a vector of protein concentrations, L, V, and U as biological constants and degradation rates, and then alpha and beta are time delays that represent the time it takes to perform some of these processes. So for example, a protein translation occurs at a rate of about six nucleotides per second. So if you take into account all these sub-processes that are involved in this, you can get fairly decent estimates of the time delays for each gene or protein represented in the model. But the most important element of the work here is the C matrix, which represents the gene regulatory network and defines gene expression based on the protein milieu in which the cell is located. So this is the object upon which the machine learning operates over to find all possible configurations of this matrix that can't be invalidated by the experimental data. So you might notice that you could also theoretically do that with the L matrix, but we end up defining it such that a single RNA then leads to a single protein. So we have multiple proteins can influence the RNA expression, but then if you have one unit of RNA expression, that generates one unit of protein. So the cells then perform various functions in response to the proteins they encounter in the environment. But the number of actions any specific cell can perform is finite, and we just determine that from the biomedical literature. So an example of a fact from the literature and how we implemented that is that TGF-beta causes fibroblasts to differentiate into myofibroblasts. And the way this is implemented is that when a fibroblast encounters TGF-beta, it, in response, produces SM-alpha through some of the equations shown previously. This then builds up into the cell, and when it reaches a critical threshold, the fibroblast differentiates into a myofibroblast, which alters the gene regulatory network and gives the cell additional functions or a different set of functions that are available to it, because the myofibroblasts, are a distinct cell type from the fibroblasts. So the most critical aspect, I think, at least in our philosophy of the gene regulatory network, is incomplete knowledge. So you don't know exactly how it works. You can only infer the rules and the influences from biological experiments and the literature, which were also informed by experiments. And so another critical aspect of that is that while the biomedical data sets are all typically looking at the same phenomenon, none of the subjects are identical. So how one person or one animal responds to a drug or injury is not going to be exactly the same as how another person responds. So the goal then of the modeling is to determine the underlying governing principles. And so I guess that gets to a point where we sometimes get a little loose with the language talking about validation. And ultimately, valid isn't the best way of describing the models that we do. So we prefer to use the term can't be invalidated. So it doesn't mean it's validated. It just means that the data you have, it's not possible to invalidate it here. So in all the work, the model structures have multiple parameterizations that are contained within the boundaries of data. So there's not one parameterization that's more valid than the others without some further, or additional experiment. So ultimately, the models are only valid in the regime of data that's been used to develop them. And I think I'd draw an analogy based on my background in physics to Newtonian mechanics. It's a very successful theory that offers lots of predictive power in a very constrained regime. So while Newton's equations of motion aren't the actual underlying laws that govern the universe, there's a large regime where they're applicable and successful and useful. And so the goal of the machine learning is to map out the space of viable parameterizations of the gene regulatory network. And there are two ways we approach this parameter space mapping, genetic algorithms and active learning. Genetic algorithms are a type of evolutionary algorithm inspired by natural selection and can be used to solve optimization problems in large search spaces that would be intractable with brute force computation. So in the genetic algorithm, a candidate solution, also referred to as an individual in the population, is represented by a matrix or vector of values, and then each value in the matrix is a gene. So you can have two individuals or solutions that undergo a simulated breeding by swapping those genes with some possibility for mutation to attempt to prevent premature convergence to a local optima. And then active learning is an efficient sampling criterion for problems when you have lots of unlabeled data and labeling it is expensive. So in this case, the unlabeled data is a putative model parameterization, and the cost to label it is to run the simulation. So the way active learning works is through uncertainty sampling. So we've got an equation for a binary classification problem on the screen. In this case, the classes would be 1, that the model parameterization fits the data or is contained within the data, or 2, that the model parameterization is not contained within the data. And so the samples that are selected for measurement, or which parameterizations the simulation runs, are those whose probability of being in class 1 or 2 is closest to 50%. So they're the ones that the current statistical model is most certain about whether or not they'd fit. And in this scheme, I first use the genetic algorithm to find regions of biologically plausible parameter space, and then we use active learning to find the boundaries of those regions. So we've just got a very brief description of the genetic algorithm here. I think most people are probably familiar with it, but in case you're not, the first thing you do is generate a random population of individuals. And to do that, we assign a random value, which we bind between positive or plus or minus 2 for each element in the matrix. And the boundary selection was semi-arbitrary, but basically we noticed that when you started getting to more extreme values, it would lead to extremely volatile model behavior, which we determined was not biologically plausible, at least for the system in question that we're using. And we then run each model parameterization with a number of stochastic replicates so we can see the boundaries of the behavior for that specific parameterization, and use that data to calculate the parameterization's fitness. And then there are two additional steps that we'll discuss in a little more detail later. The fittest individuals are then selected for breeding and undergo genetic crossover and mutation. And after that, you have a new generation of parameterization individuals. So our experimental collaborators have a number of assays for measuring protein concentrations in the animal model. And we've incorporated 13 of those into the model calibration procedure we use and we use the algorithm to minimize this fitness function. So in the model, there is a pretty decent amount of stochasticity. So any single parameterization generates a range of behavior. And the parameterization we're trying to get to with the GA is the one that generates the largest range of output that is always contained within the experimentally derived boundaries. So additionally, when we do this procedure, we also retain in an external list any parameterizations that are not invalidated by the data, even if they're less optimal according to the way we design this function. And so then after we have some of these parameterizations, we need to determine the boundaries of clinical plausibility. So we train a neural network using active learning to take as input the model parameterization and give as output whether or not that model parameterization will be plausible or successful. So to illustrate a little better how this active learning process works, we've got a visualization of a small four-dimensional synthetic data set. So 2 dimensions are shown in each panel, and then the other 2 dimensions are shown by the arrangement of panels in the matrix on the screen. So keeping that in mind, we can zoom in on the top left panel here. So the outcome space is represented by 2 colors. The red is class 1, and the teal area is a different class. And so the teal would be analogous to the clinically relevant space we're searching for. The green dots are points that are sampled some point during the active learning process, and the dark blue points are the predicted points for the interesting class. And so I think the main thing to notice here is really the density of the green points. So the algorithm samples more in areas where the outcome is most uncertain. So that's obviously going to be around the boundaries of the teal region. And the sampling density is much less sparse, or much more sparse in the less interesting areas of the panel, which is the large red area. And this same pattern exists for all the panels, and sort of as an illustration of how active learning can be used for boundary finding. The main caveat here is that the boundaries have to exist. So the more random your data is, or the more single coordinate point holes in the data, the worse or less efficiently this algorithm is going to perform. But it is very promising for spaces in which clear boundaries exist. I will say in this simple illustration, we only did it in 4-dimensional. The number of variables we're working at to optimize the model is much larger than this. So we really kind of limit it. We can't comprehensively search the space, even if you can test 1 million parameterizations per second with the neural network. When you start getting into larger parameterizations, you can't comprehensively look at the space. But from the genetic algorithm, it can give you an idea of which matrix elements have the most variability, and those are the ones we first select to learn over. So in the spirit of, or now just talk a little bit about the model calibration results, which we've done in terms of proteomics and macroscale tissue composition. And one thing to note is that neither the experimental nor the computational models go out longer than 42 days. So if you look at the figure on the screen here, this is the ratio of M1 to M2 genes, M1 being pro-inflammatory, M2 being anti-inflammatory. And you can see at this first time point, right here at day 0, this is the native tissue, M2 macrophages and M2 gene expression really dominates. And then after you injure the muscle, the pro-inflammation spikes, and then it starts slowly going down as anti-inflammation starts slowly going up. But really, for the tissue to fully return to the baseline, likely takes more than a year. We don't know for certain, but if you kind of extrapolate from this graph how long it would take for these slopes to return to their original point, it's going to be quite a while. So in the spirit of digital twins, we wanted to make a prediction for a specific individual dog. And when you make a prediction, we wanted to give a prediction cone because the concentration of a protein at a specific biopsy location can vary by a factor of as much as 1,000 over the entire surface of the wound. It's actually unclear to determine the true variance because a lot of this data, to see a lot of stuff at 0. It's not necessarily 0. It's below the limit of detection, which is typically with Luminex around 10 parts per million. So we don't really know if there's actually 0 molecule. I mean, it's unlikely to be actually 0. We don't know if it's 0.1, 1, 9, or what it is. So it's it's a little bit difficult to determine exactly, what the level of variance is. And this is something the experimental collaborators weren't super pleased with. I'm not really sure why, because the experimental data has all the same properties as the digital twin data in that, it has some of the same variance. But I think when a lot of people hear the word digital twin, I'm not really sure what they were expecting, but they were expecting it to somehow wipe away the variance they saw in the experimental data, which isn't what happened. So to do this, we took protein measurements from the dog indicated by the red dots here at day 2. So all the blue dots are measurements that were taken in some dog at some specific time point. But to create a forecast cone for an individual, we looked at these red dots in day 2, and we picked all the parameterizations of the model that contained those specific dots, none of the other dots. So we artificially lowered, the boundaries of model parameterization to here, and then ran that forward to see where they go. And you can see that it does indeed over-encompass the actual experimental data quite a bit. And this is just due, to our philosophy that we want to include everything that can't be invalidated. Now here we also did have to make a few choices when we calculated the entire parameter space about what to include and what not to include. So you'll notice that these 2 dots here in the this is actually, I believe, TNF, not IL-10. I think I have that mislabeled. These 2 dots are out of the range of what the model predicts, and that's because on this day, when they removed the bandage of the dog before they did the biopsy, it stuck. So they pulled off a little tissue, and it spiked the inflammation in that dog, or at least it pulled off tissue and inflammation spiked. We assume that's what caused it. So we didn't include that because we didn't include further wound perturbations in our model healing process. So we chose to leave those points out. So that was a little bit of a point of interpretation and a difficulty we had with the data. We weren't sure how best to simulate that. If we should try, this only happened in 1 dog that we had the bandage sticking thing. I think they came up with some techniques it was early in the experiment, so I think they came up with some techniques to limit that. But we did have to interpret a lot of the data prior to employing the machine learning algorithms to find the boundaries. So we had to determine what we want to include in the boundary and what's artificially high. So for this dog, we left day 7 for this specific dog out. So on this slide, the blue dots are samples taken from wounds that had ECM bandages applied, and the red dots are samples taken from wounds without ECM, extracellular matrix. So the shaded regions are the boundaries of behavior generated by all the viable parameterizations of the WABM with 100 stochastic replicates. And again, the zeros here aren't really 0, they're just below the limit of detection. And then the green points then represent new data. So after we trained, or after we developed the model with the ECM and we approximated the ECM as just an overall inflammation blunter, we wanted to test if the new ECM dogs still fit within the data. And they did, which was good. I think I hesitate to say that it's a failure, but it was a surprise in that, so while these regions, encompass all the parameterizations that are viable, we actually never had a single parameterization that could cover all the variation in a single dog. So one of the big surprises that we found in this work was that the intra-individual heterogeneity, so the variability in protein concentrations in a single wound, was as great as the inter-individual heterogeneity. It was as great as the difference between dogs. So due to the way we constructed the model, we weren't able to get that with a single parameterization. So each digital twin here that you can see here, and for any specific dog, is actually an ensemble of parameterizations. So I think if we wanted to make it closer to reality, we'd have to come up with a different model structure, probably with a lot more redundancy and pathways to achieve this. Because I guess I didn't mention this, even though there's significant heterogeneity in the gene and the protein expression, the phenotypes are essentially identical. So that was kind of a difficulty in the project. And then another aspect of it, one of the goals of the project was to increase the speed of healing by 50%. So actually our group weren't the people that wrote that goal. And once we got started, it wasn't even really clear what that meant, especially given the experimental results earlier on the length of the remodeling process, right? That it takes, approximately a year. So for our definition, we chose to use the length of time it takes for the wound void to fill with tissue. And there are two challenges in calculating this. And the first of those is swelling and edema, which is really the most prominent in the middle image here. So the furthest left picture was taken immediately post-surgery. So you see lots of striations and features of the tissue. All that has gone away due to the swelling in the middle picture. So that's day 7. And then the swelling has mostly resolved by day 21. So getting the model to put out visually realistic images of the wound just wasn't possible, given that we didn't know how to represent the swelling and edema. And the second challenge are the elastic forces that are present in the real world system. So when you have an intact muscle, it's always under some tension. And then when you cut it, you can see the edges of the wound pulling up, which makes the wound, you can kind of see this edge here, it goes above the skin. It makes the wound appear deeper than it actually is. So in order to calculate the volume, we first take the CT data and divide it up into vertical and horizontal slices. And then for each slice, we fit a curve using the boundaries of the slice. So you take the healthiest tissue in the scan, which is a few centimeters outside of the wound, and use a curve fitting tool to bridge the gap over the wound. So as you can see on the screen, we then recreate the surface using all the fitted slices and some filtering to smooth out irregularities. So if the curve fit to one slice is appreciably different than the curve fit to an adjacent slice, you have to rectify that. And there are two limitations of this. As I mentioned earlier, it does not take into account swelling in the center of the wound. And also, in some cases, the wound was actually at the boundary of the CT scan, so there wasn't enough healthy tissue to get a good fit. So in the figure on the right, you can see the volume of tissue. The volume of the tissue void is a function of time in some of the animals. Whoops, skipped over that slide a little bit. This shows the curve fitting procedure. In the figure on the right, you can see the volume of tissue and the void is a function of time in some of these animals. So this is what we calculated with that procedure, and it's pretty rough. Due to the swelling we talked about, the measurements between days 3 and 14. So this kind of region here is much noisier than at the beginning and the end. So in this case, we did actually fit to the mean. So proteomics and genomics we didn't, but in this case we thought the best fit line is a good approximation of mass entering into the tissue void, given all the things we left out, elasticity and swelling. And the figure on the left, you can see the total tissue in the total tissue volume in a WABM simulation. So it also includes a large volume of healthy tissue, but you can see that the wound volume fills in comparably to the experimental platform. So here you've just got a few screenshots from the simulation, starting at day 0 and ending at day 25. And the color rescales for each plot, but the yellow is the surface of the scan and the blue is the deepest region of the wound. And it fills in similarly, from the edges first and leaving a little divot in the middle until the end. It fills in similarly to what's seen in reality. So it fills the bottom a little bit at the same time as it closes the circumference of the wound. So unfortunately, the experimental considerations precluded the ability to get biopsies at the exact location that we did proteomics and genomics. So the specific cell populations and the tissue compositions, we couldn't really directly link them to gene and protein expression profiles. And so we also wanted to see and investigate if we could use the CT data to differentiate granulation tissue, scar, and healthy muscle, the granulation tissue kind of representing the initial influx of the collagens. And these are all reported to have different, appreciably different densities in the literature. So we have on screen are the CT images with boxes on them, in which the top box contains granulation tissue, and the bottom box, located, here's the granulation tissue, and this bottom box located well below the wound bed contains healthy muscle. And then we have associated histograms showing the raw CT data measuring the densities in that region of tissue. And so while there's a clear difference between the density distribution in those tissue, the difference shrinks with the size of the box, which is a problem. You can't take an individual voxel in the CT scan and determine what kind of tissue it is. And another problem is illustrated in the bottom figure, and where the muscle density pixels are green and the granulation pixels, the granulation tissue pixels are in tan. And over time, desiccation, because the wound is open to the environment, desiccation effects and scab formation alter the tissue densities such that you can't really differentiate these tissue types. So in a healthy person without a wound, without an active wound or a healed wound, it's a little bit easier to differentiate, muscle from scar and from collagen. So the wound process introduces enough additional effects that you can't determine the tissue type from the density alone in the healing wound. So as mentioned earlier, we have a large range of parameterizations, and when we train the DRL algorithm, we use a suite of those parameterizations, making sure to include many that are near the boundaries of clinical or experimental plausibility. And the motivation for this is due to the universal approximation theorem, which essentially states that an artificial neural network with a single hidden layer of a sufficient width and sigmoid activation functions, though it's generalizable to others, can approximate any mathematical function to arbitrary fidelity based on the width of the network, which I guess as an aside, sort of makes me think of neural nets as an evolution of the Fourier cosine series or something like that. But what this means is that if there's a single equation that solves your problem, the neural network is going to see through the noise to the underlying function. And as we know, that underlying function doesn't apply equally across a genetically heterogeneous cohort, and it might lose some of the generalizability. So to develop the most generalizable therapeutic policy, we try to optimize for an entire population rather than a single model. So everything we've kind of done so far is optimizing a treatment for the population rather than the individual. The specific implementation of deep reinforcement learning that we use is called deep deterministic policy gradient, which is essentially an actor-critic type algorithm to optimize a neural net that controls the simulation. And so it's also a type of Q-learning in which system states are fed into a lookup table, which tells you what action to take, or that's the classical definition of Q-learning. When the number of the potential states becomes very large, it's not practical or even tractable to use a lookup table. So instead, you use a neural network, which has been trained to approximate the lookup table and allows for a compact representation of the table and an easy handling of nonlinearities. Excuse me. So this is the type of learning that Google and DeepMind initially used to train neural nets to play Go or Atari games like Pong. It uses a discounted reward function. And what that means is that at the end of the simulation, the algorithm gets a reward, which is a measure of how well it performed. And each time step before the terminus of the simulation, the reward function is discounted by some adjustable factor. So at any specific state, the algorithm has a measure or an approximate measure of how well it's doing and an idea of what action to take that'll put it in a more rewarding state on the subsequent time step. So essentially, this algorithm is trying to guide the trajectory of the protein concentrations back to what's seen in native tissue. So here we've got just an example of two different DRL policies. One different DRL policy and the stochastic boundaries of it that we arrived at. On the left, you can kind of see the inhibition if it's less than 0, or augmentation actions if it's greater than 0, that were taken on 5 of the mediators in the model. And you can see that these policies actually did cause it to regenerate some muscle, because in the base case, you essentially regenerate no muscle. So any improvement is good. We weren't able to eliminate the collagen, which was a little bit of a problem. And so I think it sort of invalidated the hypothesis of the whole experiment, was that just by manipulating M1 and 2 or pro and anti-inflammation, you could generate a beneficial healing. And the reason for this is that pro-inflammation prevents significant muscle growth, but anti-inflammation augments fibrosis. It activates fibroblasts, it causes them to secrete collagen, and it causes that to be remodeled into scar. So I think one of the results of the project was that the hypothesis of just manipulating inflammation wasn't going to work. The other result that we demonstrated with the model, and I think ultimately convinced people was true, was that if you want to have a beneficial healing phenotype, you can't increase the speed by 50%. Because when you look at the healing now, essentially a bunch of cells come in and they just dump collagen into the wound in a chaotic and disordered fashion. They just fill it up as quickly as possible. So it's something that can happen relatively quickly compared to a muscle fiber, which has to build up one cell at a time. One cell has to fuse at the end before the next cell can fuse. So it's naturally going to take longer. So this was actually a success of the model that we were able to demonstrate why this goal of 50% wound closure speed up, as well as eliminating fibrosis, was unachievable. You could speed up the closure of the wound by speeding up fibrosis, but that wasn't really what was desirable. So the validation of our predictions, the effects of ECM could be well predicted by approximating the ECM effect as an inflammation blunter. So we recognize that the wound interface, the surface of the wound with the environment, causes an ongoing inflammation. We simulated or approximated the effect of ECM by just turning down that inflammation significantly, which was experimentally validated. Unfortunately, we were not able to test any of the protein-based treatments due to experimental limitations. So in the slide where I talked about the overall goal of the project, the AI informed some genetically engineered cells what proteins to make and when to make them. The protein delivery turned out to be significantly more complicated than expected. So essentially, proteins were put into the wound. They were not absorbed into the tissue, which is not what we expected. And thus, any protein application didn't really alter the biology in any statistically significant way. So to go further in this project, we definitely need to come up with a different delivery method for the therapeutics. And then just a little bit going into the future work. So we are still working on this. Right now, it requires sensors to measure features that aren't typically measured clinically, at least not in a real-time fashion, the molecular mediators. But going forward, we want to include clinical measurements as functional outputs of the model. So things like blood pressure, heart rate, the ability of the kidney to filter out creatinine, and that kind of thing. So ultimately, we're going to need to link molecular time series data to clinical observables. So in future work, we're linking these high-resolution computational and mechanistic models of organ systems or muscles to a physics-based model of the cardiovascular system. So we're using the biogears, which I think has been talked about definitely in the IMAG group. I don't know in this specific seminar series. And we're linking that up with the delayed differential equations in the model. So we went ahead and that's how we inform the time step of our model to make it match up with biogears. That way we can have a higher fidelity representation of inflammation than is available in the biogears package, but also then link that with physiology. And so I guess I just kind of want to end with a quote from maybe even someone on the Zoom call. I heard this at dinner when we were at the meeting in France a couple years ago. And this guy said, biology is where physics was 100 years ago. And I really like that because as really became apparent to us during this project, we're still searching for the correct mathematical representation or the correct computational representation to make a lot of these things clinically and experimentally useful. And so I think while we've made progress on that towards that area with this project, it's certainly not a solved problem, and the experimental and data difficulties we encountered here were unexpected and have given us a lot to think about. So with that, I'm happy to entertain any discussion.
So we will go ahead and open the floor up for discussion.
Well, I might start with one if nobody has one. Well, first of all, thanks you for the talk. It's very, very nice, very great work. Many aspects you covered. You mentioned you have there is a part of ABM in your idea, and I wonder if there is a specific mechanistic model under the ABM, because typically we have rule-based or mechanistic model underlying ABMs, or if instead, well, you have chosen a different approach. If you can discuss on that, that's
Yeah, so there is a specific rule set or underlying structure to govern the model. It's just that we allow many parameterizations of those rules. So the way we structured the model, there was no single parameterization that could cover the entire space. And the reason for that was all the zeros and the huge range of variance. So it may be possible to construct it's certainly possible to construct a mathematical representation that can do all of that. I think it's much more complicated than what we did, which is already pretty complicated. But yeah, it's a single rule structure with varying parameterizations of that rule structure. But like cell behaviors are predefined, so the cells can't ever gain new functions, but they can perform those functions in response to different concentrations of proteins based on the individual parameterization.
And then perhaps this is too technical, so I'm not asking you to go in greater detail. How do you plug in a mechanistic model, or if you have used specific framework? That's one question. And how you do plug into the something more statistic based or AI based?
So we didn't use a
Find the
specific
two.
framework. It's all just custom made in C++. To interface with machine learning, we use a Python library called ctypes. So that allows Python to read memory from a C++ program. So we can use all the machine learning aspects of Python while, concurrently while the simulation is running, and step the simulation and we can do it as often as we want. So the Python can then step the simulation a single step forward, run whatever machine learning calculation is going to do, then step it forward, or it can run the whole thing and record the data. So we use C types to link Python for all that.
So essentially, the mechanistic model gets informed by the AI at each time step, and it continuously adapts.
Hey, I asked a quick question in the chat
Oh,
on
sorry.
how you were initializing the cell populations in the system. So how did you get the proportions of cell types that you needed?
Well, we initialized it just by assuming that everything that still existed was healthy muscle. And then from there, it generated the inflammation caused fibroblasts to generate the granulation tissue. So at time point 0, it's probably not exactly what it is. And due to the nature of the biopsy, it actually, once the wound starts healing, it never penetrates really deeply into the healthy tissue. But the tissue inside the wound void is all validated quantitatively, the histology. But the tissue that's outside of the wound void that generates the behavior isn't measured experimentally.
Okay, so were you using like transcriptomics or something, or RNA-seq to get the specific cell types that you needed?
Yes.
Okay.
I was not. Our collaborators at Pittsburgh did that, but yeah, then they gave
Okay.
us the data. Yeah.
Okay, thank you.
I think if there aren't any other questions, we can go ahead and bring this to a close. Thank you again, Chase, for a
Yeah,
great
you're
talk
welcome.
today.
Thanks for inviting me.